Chapter 10, Part 4. This part will include meiosis second, detail of meiosis second. Then we have to differentiate how does it differ from mitosis. And in the last, we have to differentiate mitosis and meiosis cell division. This is Part 4, Chapter 10. Yes, this is Chapter 10, Part 4. Already we have discussed meiosis first, different stages of meiosis first, that is prophase first, and detail of prophase first, which was very important one, that is substages leptotin, zygotin, pectitin, diplotin, and then dikinesis. And you have seen in this stage is some important, that is changes that was leading to recombination of, that is genetic constitution. Now we have to study meiosis second. As I told you, meiosis second, that is, it is almost equal to equational division, that is mitosis, and also known as homotypic division. It resembles to mitotic division. Why? Because number of the chromosome that will remain equal to parental cell. No need to say here, that is, this was the 2n. It has divided into, that is first meiosis first, reduction division was there, daughter cell was having n and n number of chromosome. Now here meiosis second will start. This will divide, the two cells will be formed, the number of the chromosome in daughter cell will be equal to this parental cell, and as a result of this, that is, there will be 4 haploid nuclei, haploid cells will be formed. Right? So meiosis second is necessary because each daughter cells formed from meiosis first contains chromosome, each having 2 chromatid with 2C amount of DNA. Reduction in DNA from 2C to 1C occurs in meiosis second only. And then in interphase, again this amount of DNA will be doubled. We have to see detail of this. What is there, children, in meiosis, that is second? The first stage here is prophase first, initiated by, that is just after cytokinesis, if it is there, that is meiosis second, prophase second will start. What is there? That is before this, the chromosome are fully elongated. In contrast to meiosis first, meiosis second resembles to a normal mitosis, nuclear membrane that may disappear if it has reappeared in cytokinesis. Usually it remains disappear, and then there is no need that it will disappear now by the end of prophase second. Chromosomes, again, they will become compact. Metaphase second, what will be now? See here in metaphase second, at this stage, chromosomes are aligned to equator. Remember, if this is the cell, dear children, then the chromosome, these are aligned to the chromosome, that is equator plate, like this. And each chromosome is having its chromatid. There is only one row of chromosome. Each chromosome is represented by its chromatid. This is known as metaphasic plate, one metaphasic plate. And the same as I told you earlier, that is this centromere, both sides, it is having this kinetochore plate where this spindle fibers will attach, and in anaphase second what will happen? What will go? That is, this is the chromatid which will go to the opposite pole, not the full chromosome. Therefore, if number of the chromosome we have taken here, that is, let us say 1 and 2. So here also, that in daughter cell, number of the chromosome will be 2. Remember this thing, number of the chromosome is equal to 2, but here the chromosome is represented by single chromatid, while here the chromosome was having 2 chromatids. Is it right? And that is, eventually this amount of DNA will also be halved here. So you see here, that is, here microtubule they will make, that is spindle rays, and spindle get attached to the kinetochore of the sister chromatid. You can recall mitosis, that is metaphase. In anaphase, I told you, it begins with simultaneous splitting of the centromere. Here that was centromere, one centromere. Now here the centromere will split so that each chromatid or so-called chromosome will have its own centromere. Okay, and allowing them to move towards the opposite pole, because the microtubules, these microtubules, they will shorten and they will draw the chromatid, so-called chromosome, to the opposite pole. Is it right? Now you see, telophase second, let that was the parental cell having 2n number of chromosome. That is, this was the meiosis first. That is, two daughter cells have been formed. Two daughter cells, let this is 1 and 2 chromosome, that it was having 4 chromosome. Now the daughter cell is having, that is 2 chromosome here, 2 chromosome. This is 1 chromosome, 2 chromosome. The daughter cell is having, that is half number of chromosome. What happens now? In prophase second, this is over. In metaphase, both these they have been arranged on metaphasic plate, and these chromosome, these are held together by this, that is centromere, metaphase. One metaphasic plate we have here. This is one, that is metaphasic plate, right? And here the centromere, that is, it held the two chromatids of the chromosome together. What is there in anaphase now? Yes, anaphase, this centromere will split so that each, that is chromatid, will have its own, that is centromere. This chromatid chromosome that will split into, that is centromere. So here this is one centromere, another centromere, and this spindle fiber, these are attached to this centromere, and they will pull these, that is so-called chromosome, towards the opposite pole. How many number of chromosome we have here? 2. The chromosome is having how many chromatid? 2 chromatids. Each chromosome is having 2 chromatid. Here also the number of chromosome to this side, in daughter cell also 2 chromosome and 2 chromosome. But what's the difference? Here the chromosome is represented only by one chromatid. This is, it is the one, yes, this is one chromatid. Is it right? And now the chromosome, so-called, consisting of only one chromatid, I repeat many more times, they have reached to the opposite pole. It will be followed by cytokinesis here, so that 2 daughter cells will be formed from each. The 4 haploid cells will be formed. Each is having how many number of chromosome here? Number of chromosome is 2 and 2. That 2 is the n number of chromosome, n, n, and n. The parental cell, this is the parental cell of this, it is also having 2 number of chromosome. But this is the grandparent cell which has gone under meiosis, it was having 4 chromosomes. So this is meiosis first, and that will be, that is meiosis second. Is it right? Any problem in this? Okay. Now we switch on to, that is cytokinesis. As I told you, there also cytoplasm that divides forming furrow formation in animal cell and cell plate formation in plant cell, and after cytokinesis a single cell divided into 2 daughter cell, that is the parental cell, that has divided into 4 daughter cell. Significance of meiosis, see now. Yes, gametes are formed through meiosis, and gamete formation is essential for sexual reproduction. This part is very, very important one. You have to, that is, pay extra attention to this. Meiosis maintains fixed number of chromosome generation to generation. What we mean by this? Let the human male is having 46 chromosome. The female is also having 46 chromosome. How meiosis maintains definite fixed number of chromosome? You know that they will, in doing sexual reproduction, that will divide. Ultimately 2 cells will be formed having 23 and 23 number of chromosome. Now again they will divide, so that 23, 23, 23 and 23 number of chromosome will be formed, and later on they will be converted into sperms. So here the gametes have been formed. Almost similar process will happen there in female, that it will divide, 2 cells will be formed, 23, one will be smaller one. Again it will complete, 2 cells will be formed. This is the functional ovum. The 3 cells will be rejected. This is gamete formation. So meiosis has reduced number of chromosomes, and in fertilization that it will get fused with this, again 46 number of chromosomes will be restored. Are you getting my point? So means meiosis maintains definite number of chromosomes. If the meiosis will not be there, after every sexual reproduction, number of the chromosome will be doubled. Meiosis is the main cause of variation. You see here, it's meiosis. This is the cells have been formed. These cells have been formed. So here there was what crossing over occurs. Crossing over means there is what recombination of genetic constitution. So recombination of genes that is responsible for that is variation. And now very important point we have here, that is children, you see meiosis, 3 types of meiosis are found. The most primitive one is known as, that is zygotic meiosis. Zygotic meiosis also called initial meiosis. What is there in this zygotic meiosis? That the main body of the organism is haploid N and N. This is the main plant body. What will be there? That they will produce the gamete having N number of chromosomes. That this is male gamete, this is female gamete. Now they will fuse, the zygote is formed, and now there will be meiosis in zygote, and result will be, that is again this N stage will be stored. So this zygotic meiosis is found in case of, that is lower plants, like that is algae and that is fungi. Can you give answer of one question which is a very important one? That is meiosis always results into haploid cell. This is right or wrong? Yes, this is right. Haploid cells are produced by meiosis. 1. The second is meiosis will always be in diploid number of chromosomes. The cell that is in diploid cell. Yeah, this is also right. Meiosis will be only diploid cell. See the other thing here. Here the gametes have been formed. The question is, the gametes are formed only by meiosis? No. Here you say gametes are formed by mitosis also. These are the gametes formed by mitosis. Is it clear? Other type of that is meiosis is that is known as sporogenic meiosis. Sporogenic meiosis is found in case of plants, remaining plants, bryophytes, yes, pteridophytes, gymnosperms and angiosperm. As a result of this, they make the spores. That's what is known as sporic meiosis. And gametic meiosis is found in case of animal known as terminal meiosis. As a result of this meiosis, the gametes are formed. This is found in case of, that is animals. Is it right? Come to the next now. Now children, we have to differentiate. You have seen the detail of mitosis and meiosis. We have to differentiate mitosis and meiosis. Some general points we can discuss. It is found in somatic cell known as somatic division, also known as that is growth division, because of this mitosis, number of cells increases. It is found in reproductive cells known as that is reproductive division. Mitosis is found in diploid and haploid, but most common this is in diploid. It also found in haploid, as I told you right now. That is in case of lower plants. Yes, that is mitosis will occur in haploid cell, but the meiosis is always found only in diploid cell. Okay. Come to the next now. Mitosis, it produces 2 cells. will divide, 2 cells will be formed, while meiosis will make 4 cells. The 2 successive divisions are there, and in that way the 4 cells will be formed, and all these 4 cells will be the haploid one, having 2 phases, one and the second phase. It doesn't introduce any variation because there is no recombination. It introduces variation because there is what recombination, or we can call that there is, that is crossing over is there. As a result of crossing over, there is, yes, that is variations arise. Number of the chromosome, same as in mother cell, equational division this is called as. Number of the chromosomes are equal to parental cell. Equational division, here these are being reduced. It is required for growth, repair and healing, and this is required for sexual reproduction. Important. Come to the next now. This is prophase. It is simpler. It is very much complicated. It has divisions: leptotin, zygotine, pectine, diplotin, and that is dikinesis. Dikinesis. This is complicated one. Bouquet stage and synapses are absent. You know the detail of this. That is in zygotine, what in leptotin, that is all these chromosomes, these are attached to the that is nuclear membrane, leptotin. In zygotine there is, that is pairing of the homologous pair of chromosome. In pectine there is what? The crossing over is there like this. That is, it is found. Chiasma formation is there. Metaphase, centromere, that is here. In metaphase what is there? The chromosomes are arranged on equator. This is centromere. Centromere is present over the equator, right? And metaphase is there. Arms are facing towards the pole. Arms are facing towards the pole. It will be like this, that these are the, that is 2 chromatids. Centromere project towards the pole. What is in anaphase? Centromere that divides important. Here, that is centromere will not divide. The 2 chromosomes, these are held together, and actually that is this complete chromosome that will be withdrawn towards, that will be pulled towards the opposite pole. Chromosomes, that is here, that is represented by single chromatid. In anaphase, that is, this is one chromatid like this, this is another chromatid, while here complete chromosomes will be pulled towards the opposite pole. 2 chromatids of a chromosome separate and becomes a daughter chromosome. Here chromatid do not separate. At anaphase, chromosome moving towards the opposite poles, these are similar, these are dissimilar because of, that is crossing over that was there in prophase first. That's why the chromosomes, these are dissimilar in genetic constitution. Now, telophase, it is essential, in which the nucleus and nucleus membrane reappear. It may or may not be present. It may be absent. Here, what is there? That is, this is, that is 2 chromosomes, 1 and 2, 1 and 2, 2 chromosome here, 1 and 2. That is not necessary that a telophase may be present or cytokinase. Telophase first may be absent. That is, these chromosomes will remain without development of the nucleus membrane. Because in time to come, in meiosis second prophase, again this nuclear membrane has to disappear. Therefore, just to save energy, there is no need that this nuclear membrane will appear here. Cytokinesis is essential. Daughter cells possess same genetic constitution. That is, that may be or may not be here, because if this cytokinesis will appear, then that is may or may not be there. Not compulsory. We have to see, discuss. That is okay. 4 daughter cells are formed. They are different in their genetic constitution. And now we have to see differences between mitosis and meiosis first. Differences between mitosis and meiosis. In the beginning we told, and we have realized, that in mitosis what is there? That is, that this is the n number of chromosomes. The daughter cells are formed. They are also having n number and n number of chromosomes. Number of chromosomes are equal. Here also you see, that is, that was the meiosis first, having 2n number of chromosomes. meiosis first, two daughter cells have been formed having n and n number of chromosomes. Now this is meiosis second. For example, in this cell, the two cells will be formed having n and n number of chromosomes. So it appears that this is equal to this, that is mitosis and meiosis second. These are almost equal. But even then the differences are there, and the differences, children, are very important one. An interphase prior to mitosis. Interphase. This mitosis is there. You see, that is here is interphase is very much essential, that before it enters into this M phase, there was G1, there was S, and there was G2. So this normal mitosis, before it enters into M phase, interphase is very much essential. Here interphase may or may not, because it has been formed right now. So just after completion of the meiosis first, there is no need of interphase. What happens in interphase? That is, there will be decondensation, that is condensation, and then disappearance of interphase. What is there in interphase? There is synthesis of, that is, DNA material, duplication of DNA will be there, and all this material required for, that is, mitosis or that is M phase will be synthesized. There is no need of that. Meiosis second, is it? If it is present, that will be known as interkinesis. Here is S phase is essential, because here, that is, DNA duplication is very much essential. Here, because the DNA of this have been halved here, so at this stage, that is, S phase is completely absent. That's why it is said that is meiosis consists of two, can you summarize? Two karyokinases. It is having two karyokinases and only one time, that is DNA duplication. One time DNA duplication. Here it has only one time karyokinasis and one time, that is DNA duplication. S phase is there, is it? Daughter cells formed after mitosis, they resemble to the mother cell. There is no, that is, genetic variation. But here, these daughter cells which have been formed here, because one different type of chromatid will go to this, different type of chromatid will go to this. That's why these daughter cells are not similar to this cell. They may differ from mother cell quantitatively, and that is qualitatively. Is it right? So these are the basic and major difference. And you see, that is numerical problems related to amount of DNA, number of chromosomes. This is very important question. I told you, in meiosis there is two time karyokinasis, only one time, that is DNA duplication is there, while in this case only one time karyokinasis, one time of DNA duplication is there. Is it right? So you have to see differences. We can have a very fast recap of this part. Yes, see now. Yes, meiosis second. This is almost similar to mitotic, but the differences are there. Prophase second, metaphase second, anaphase second. How does it differ from, that is, prophase first, metaphase first, and anaphase first? That detail already we have discussed. I told you here that this is the diploid cell which has undergone this meiosis. 4 chromosomes we have taken here. The 2 chromosomes will be here. That is 2 chromosomes. This is 1 chromosome. This is another chromosome. This is 1 chromosome. This is another chromosome. Chromosomes here are represented by 2 chromatids. And when this will divide, that the daughter cells, number of the chromosome will be equal to this cell, but here it is represented only by 1 chromatid. Very important. Amount of DNA in this, if this amount of DNA is 2C, number of chromosome is how many? That is 2. In this we have, that is DNA amount, that will be how much? DNA amount will be 1C, and the chromosome number, yes, chromosome number will be again 2. But what's the difference? Here these chromosomes, That is, these are always 2 chromatid. Remember, in numerical problem you have to be very careful. But here, this chromosome, it has only, that is, 1 chromatid. Is it right? So this is part 4 of chapter 10. Thank you.